So I just wanted to get started here and give you guys a little bit of a repeat from chapter 7. So I'm going to start here with sharing the content, and I'm just going to be using a whiteboard here. So chapter 7 is all about membranes, and I would remind you from chapter 6 that or actually from chapter 5, that I told you need to tattoo on your brain that all membranes in all cells are made of a phospholipid bilayer. And I wasn't joking about that. Or you can say composed, that sounds a little more scientific, of a phospholipid bilayer. And all cells, right? All cells, prokaryotic cells, eukaryotic cells, all of them are composed of a phospholipid bilayer. And I would remind you that the phospholipids, we looked at those also in chapter 5 as a specialized type of lipid. What we mean by bilayer is that there are 2 layers of these phospholipids. Now, as I'm drawing them, okay, the head of the phospholipid, this is the portion that has the phosphate group, PO4 I'll put, and then there's a choline group on top of that. And all of this portion is negatively charged. These tails, these are lipids, and lipids, this is the fatty acid chains here. The reason that this tail has a bend or a kink in it, and it looks like someone, like a person almost, that is giving a kick, is because there is a double bond there between 2 of the carbons. So this would be considered an unsaturated fat. The phosphate heads, we call them hydrophilic, which means literally water loving, phile for love and hydro for water loving. Because they're negatively charged, water being polar covalent, right, has those positively charged hydrogens that are going to point toward the phosphate head. Outside the cell and inside the cell, because inside the cell is predominantly water. It's between 90 and 85% water. This portion we refer to as hydrophobic, or water-fearing, phobia, because this is the part that is the lipid, and lipids are non-polar, polar covalent bonds. And because they're nonpolar, that means no charge, and so they don't want to be near the charged water molecules. Erase all that so I can add some other components here. So we start with, again, a picture of the components of the membrane, because we always do structure first and then function. And the structure of the membrane is composed primarily of phospholipid bilayer, proteins, and some sort of a sterol or steroid. In the case of animal cells, that's going to be cholesterol. So let's give this a draw here. So I have, first I'm going to make the phospholipid bilayer. So I'm going to make my phosphate heads and my lipid tails. And again, it's a bilayer. In your book, the phosphate heads are gray and the tails are like a yellowish color. Okay. And then I'm going to make my proteins blue because I don't have purple on this particular device, but I do have a nice bright blue here. And so that odd-shaped blob sort of structure that I just drew, that's supposed to be a protein. And then there would be phospholipid bilayer would continue. And then as in your book, I'm sort of taking a look at that picture 7.3. There might be another protein here, kind of blobby shaped, with like some attachments, and then maybe a different compartment right there, right? And in this case, then we would see phospholipids, right? They form all around the protein. They fill in all the spaces. And even though I'm drawing this in 2 dimensions, you have to try to remember that it's a round cell, the whole organism. There probably wouldn't be that much space between those. There we go. So we have phospholipids here. We have some proteins that I put in the membrane, and then I'll make the cholesterol molecules yellow. I can't draw them in the exact correct shapes in your book where they completely redraw the cholesterol molecules. So I usually draw them as 4 squares, kind of like this. So that would be my version of cholesterol in the membrane, which again, you might remember from chapter 5. It's 4 rings with a chain of fatty acids. And I'll continue drawing a few more phospholipids here. Hydrophilic heads pointing outside the cell and inside the cell. Proteins in blue, and these steroids, and in this case, like I mentioned, the steroid in animal cells would be cholesterol. And the main function of that cholesterol is to give the membrane a little bit of rigidity, so it's not so super squishy, if you want to think of it that way. Okay, now let's talk, we talked about the phospholipids, let's talk a little bit more about these proteins. Any of the proteins that go all the way through the membrane, like this protein here, right, like this guy, we would call that one an integral membrane protein. You might also see it called a transmembrane protein in later in our book, or in a different textbook. If you're reading a different biology book, they might call it transmembrane protein. And functionally, many of these are also going to be carrier proteins, which we'll talk about a little bit later. This type of protein is a little different. See how it's only on the inside of the cell? And this one too, it's only partway through the membrane. It doesn't sit all the way through the membrane, right? So these membranes would be called peripheral membrane proteins because they're on the periphery or on the edges, okay? And again, we already spoke about the cholesterol. Some of these proteins are also going to have extensions on them. And I'm going to try to draw those kind of like, so they do kind of look like glucose. They might have a chain of glucose molecules on them, and then we would call them a glycoprotein because they have a carbohydrate attached to them. Just put CH2O there to remind you. Some of them might have a lipid attached to them. Some of them might have another protein attached to them. In your book, it kind of looks like a little arm sticking out. And that protein would then bind to the fibers in the extracellular matrix. And those are also proteins. So I'll draw them kind of like tubes here, fibers going around, right? So that might be fibronectin or collagen, right? And again, this picture you would see if you're looking at chapter 7, and you would also see it sort of at the end of chapter 6 when we were talking about extracellular matrices. Now, this again is the membrane that I'm drawing here is an animal cell membrane primarily. It is not so much a plant cell membrane. A plant cell membrane would have a different steroid in the membrane, as well as a different extracellular matrix. It could be, you know, a cell wall made out of cellulose. Now, the next part about the structure that I would remind you, and you'll see this term in your book as well, your textbook refers to the membrane as a fluid mosaic model. And again, that means just like it sounds. The mosaic part is to help you remember that there are different components of a membrane. The phospholipid bilayer, many different proteins, and the steroids or sterols in that membrane. The fluid part, that pertains to that these proteins they can actually move through the membrane, or the membrane is a fluid system in that the proteins, like today, right at this moment, you might look at this protein here, and it's located at this particular spot. 2 hours from now, it might be over here, going the opposite way. There's a nice picture in your book that shows an example of one of the experiments they did to show how the proteins move in a phospholipid bilayer. And it's figure 7.4, and I talked about it earlier today as well. Most of the time, I would remind you, most of the time, the proteins in the fluid mosaic model, the proteins are going to be moving what we would say laterally. So they're going to be able to move in this direction, or if we were trying to look at this sort of three-dimensionally and we could see the other phospholipid heads back here, the proteins would be able to move back and forth on the surface of the cell, right? They usually, they rarely flip. And what I mean by flip is this side of the protein, like here where I've highlighted in red, rarely changes places with the inside and vice versa. That usually does not happen. I'm going to erase that. Yeah, took out my whole protein. Let me draw him. Oh, extension. There. Okay. I think that's all I need. One more thing that I wanted to tell you and remind you about the structure of the membrane and the fluidity of the membrane is that membranes will fuse. So when we have fused, I'll say easily. So when you have, for example, a vesicle inside a cell and it literally bumps into the cell membrane, they just fuse together and the cell membrane enlarges and gets bigger. You can see some of those pictures if you look back to chapter 6 as well. Okay, I'm going to start another whiteboard here to begin to talk about the function of the membrane. Right, so always it's structure first and function second. The function of the membrane can be simplified or abbreviated in a single set of words. The membrane is the selectively permeable barrier to the cell. It is a selectively permeable barrier. Okay, so let's just take that word by word, right? Selectively permeable. Some molecules can permeate or move through the membrane, and some can't. The ones that can't, right, that's why we're saying it's selective. Not every molecule can move through the membrane. And it is the barrier to the cell, right? It's the edge of the boundary of the cell. It's the, the edge of the yard, the edge of the body of water, whatever you want to think of it. It's what delineates a single cell from all other cells, okay? Okay, so now, how do things move through this barrier, right? It's selectively permeable. Which things can get in and out, and how do they actually cross the barrier? How do they move from inside the cell to outside the cell, or outside the cell to inside the cell? And this occurs mainly in two forms. We call it passive transport or a molecule will move by active transport. And we have to understand these first two words before we can talk about specific types of passive transport and active transport. All the rest of the chapter is about different types of passive transport and active transport, and we're going to go through them one at a time. So let's start with passive transport first. Okay, passive transport, passive, that part of it, is referring to the fact that no ATP is required. Now, I would remind you that ATP is the form of chemical energy for the cell. So no ATP, or you can say no energy is required. Why is that? Because molecules are moving, say, down or with the concentration gradient. Another way to say that is molecules are moving. What we mean by concentration gradient is molecules are moving from a high concentration of that molecule toward a lower concentration of that same molecule. So when I use these square symbols like that, those square parentheses, that's a shorthand from chemistry. It stands for the concentration of whatever molecule or chemical element that I put inside that. Okay, so this means concentration of. Okay. The up arrow means a high concentration. The pointing down arrow means a low concentration. And this arrow from left to right, that's indicating toward in this case. Okay, so this is all about why we say how we describe or define passive transport. These two parts. First, no ATP is required. Why is no ATP required? Because the molecules are moving down or with the concentration gradient. Each molecule moves according to its own gradient from a high concentration to a low concentration. And I'm putting this in red on purpose until the molecules reach equilibrium. Equilibrium, which means the same concentration on both sides of the membrane. Okay. Okay, now let's take a look at some examples of passive transport. Give you a minute to copy that down, or I guess you can probably freeze your video and watch it. I'm just going to erase. And again, like I said, let's take a look at some examples of specifics of passive transport. So I'm going to put passive transport back here. And I do think I'm going to go to a new board. Let's go back. So again, when I write that down, this is a way to help you study for this material, for this exam coming up. When I write that down, even when I just wrote passive transport, first thing I think of in my head is no ATP required, molecules moving with the concentration gradient. And I think of where are they moving? Across the membrane. Why? Because the membrane is selectively permeable. Okay, if you run through all of those items in your head each time you write the word passive transport, that's helping you remember how they're related to each other and how they're joined together and how they're relevant. Okay, so let's take a look at the first type of passive transport. It's just called diffusion. Again, sometimes you'll see this called simple diffusion. And diffusion is just that. It is movement of a molecule across the membrane from a high concentration of that molecule toward a lower concentration of the same molecule. So example, what can actually move by diffusion? Mostly gases. So think of oxygen, carbon dioxide, nitrogen, those are all gases, right? And if I were to redraw my phospholipid bilayer here real quickly, put a protein in there. This would mean oxygen. Let's say there's lots of oxygen molecules outside the cell, and there's only 2 inside the cell. So there's higher concentration of oxygen outside the cell than inside the cell. So that oxygen is going to move through the membrane until equilibrium. Passive process, no ATP required, solely based on where there is a higher concentration of the molecule compared to where there's a lower concentration of the molecule. Diffusion. Simple, like I said, sometimes you'll see it called simple diffusion. Another type of diffusion is we refer to as facilitated diffusion. And that means diffusion with help. And the facilitator in this case is going to be a protein. And it's going to be an integral membrane protein that spans the membrane from one side to another. And sometimes they'll be called channels or carrier proteins. Because usually they have a like a tube in the middle, so that there's a special place for a particular molecule to fit. So I'm going to continue to draw my phospholipids here. Try to get rid of a little bit of this so I have some room to put a different molecule. Put the protein in here. And this protein is going to be drawn like that. And I'm just, okay. Okay, this protein is going to be my example of a carrier protein. So these for facilitated diffusion, these usually are for molecules that are a little bit larger, maybe the building blocks, or what we're calling the monomers in chapter 5. Sorry, chapter 5 never goes away. Bummer. Things like a monosaccharide, maybe a glucose, or maybe an amino acid. I usually just abbreviate amino acids like that, A.A for amino acid. So I'll make it look like glucose here. Let's see. Better yet, let's just make it a triangle. Something simple. Okay, so let's say these triangles are to represent glucose. Higher concentration outside the cell than inside the cell. Diffusion. The molecule is going to move from where there's a higher concentration to where there's a lower concentration. So I'll put a few here inside the cell. These glucose molecules are going to travel right through this carrier protein. And this carrier protein is specific for a single molecule, specific for just glucose, for example, just triangles. If I have squares out here or circles, they're blocked. they cannot get through. Okay. There is a nice video, one of the bioflicks that I assigned in chapter 7 is about facilitated diffusion. So you'll see a nice picture of that. Okay, I'll pause for a minute while I erase this. And then the last type of passive transport that we need to talk about. So this is the third example, the third specific type of passive transport. And again, as I wrote that, I remind myself, no ATP required, moving from high concentration to low concentration. And so number 3 is a process we call osmosis. And that is the passive transport or the diffusion of water molecules across the membrane of a cell. I'll put cell membrane. Okay. Why are they diffusing? Because they are moving, it's passive. They're moving from where there's a higher concentration of water toward where there is a lower concentration of water. Simple as that. Okay. So if I have my cell over here real quick. Right, with my proteins in it and my cholesterols in it. Right. And I'll make the water molecules blue as well. If I have my water molecule here, and that's the oxygen and the 2 hydrogens, right? The oxygen and the 2 hydrogens, and there's more water outside the cell than inside the cell. These water molecules are going to diffuse through the membrane again until equilibrium. Because this is a common occurrence and a common stress on cells, how much water is inside, how much water is outside. And it's related to the pressure of how much water is inside versus outside. We usually discuss at this point different types of solutions that cells may find themselves in. And this is about where we lost connectivity today when we were talking about hypo and hyper and isotonic solutions. So I'm going to draw a beaker and put a solution in the beaker. And in this first example, the beaker is going to be filled with pure water. And into this, we have a cell, which I'm drawing extra large so I can draw inside of it. And inside the cell, of course, there are all the proteins, all the organelles, various ions. There's all kinds of what we would call solutes inside that cell, right? The water is the solvent. And there might only be, let's say, 80% water inside this cell. And what happens then is that puts pressure on the cell membrane because we have a higher concentration of water outside the cell than inside the cell. So the water is going to move from high concentration to low concentration. It's going to move into the cell until it reaches equilibrium, if it's possible. Okay. The cell, if it does not have a mechanism to pump that excess water back out, it will swell. And if the pressure is great enough, it might burst. Or we refer to that as lyse. This solution has a name based on the pressure that it exerts on the cell. We call this type of osmotic solution a hypotonic solution. And how I remember it is hypo with the O, hypo, more water outside than inside. It's going to cause my cell to swell. Now, if I have a solution that is perfectly the same inside the cell and outside the cell, and let's say out here there's 80% water as well, because we have the same solutes in the solution, let's say salt and glucose, some amino acids, right? If it's perfectly matched, the water is still going to move, but the water will move one molecule out, one molecule in. And that solution we refer to as isotonic. It's the same tonicity or the same pressure of the water inside and outside the cell. And the last solution that we would draw would be one where, let's make this like 70% water outside the cell. There's 80% water inside the cell. And again, why is this 70%? Because we've dissolved lots of solutes into the water, into the solution. So in this scenario, there's more water inside the cell than outside the cell, so the water will travel in this direction. The cell will shrink, shrivel, whatever you want to say. This type of solution we refer to as a hypertonic solution. Now, the hardest part for students most of the time is that these words, hypotonic, isotonic, hypertonic, especially the hypo and the hyper, we can use that to describe either the solution that we put our cell into or the solution inside the cell. In the case of this particular scenario, this particular beaker, the solution is hypertonic. The cell would be hypotonic relative to the solution. So always the solution that has more water, right? So it's a relative term, more water on one side of the membrane compared to the other side. Whichever side has more water, that's the hypotonic side of the membrane, and the other side of that membrane is the hypertonic. And that is going to cause water to move in and out of cells. All right, let's get rid of that. That was our last type of passive transport. And I think that's where we're going to stop today. And then tomorrow we will continue with active transport.